Hello aspirants, welcome to another part of 𝙂𝙎 test series in GDC online classes. This is 𝙂𝙎𝙎𝙎𝙄𝙊𝙉, your cognitive faculty, and we have discussed part 1 for this 𝙂𝙎 test series on pharmacognosy subject, and this is a part 2. So where we are going to have same MCQs or same type of MCQs on another uh, part which includes the same introduction of pharmacognosy. We're going to see other aspects or other important topics in pharmacognosy introduction, where we have came across like so many concepts of introduction, history, origin, and like how the rest of the important topics will be discussed with a very neat explanation in the form of MCQs. Clear? Are you ready, my dear students? Yes. Without getting delay, we'll start the session. Welcome to the part 2 𝙂𝙎 test series of pharmacognosy. Clear? Yes. Let us have the first question itself. Yes. In the first question itself, we have this: select the incorrect group based on the chemical classification. Select the incorrect group based on the chemical classification. We have different types of classification, but they're asking about to pick the incorrect group based on the chemical classification, which means the all the options are arranged, okay, except one option. All the options are gave in a chemical classification, or else only one is incorrect. Okay, so we'll see digitalis ergot �々𝙬𝙗, naxomica ergot cinchona datura, peppermint clove eucalyptus and garlic, digitalis senna cascara and licorice. Yes, do post your answers. Yes, we'll have four options. From that, which one is incorrect group according to the chemical classification? We'll see the right answer so that we'll have a brief explanation about the question. Yes, digitalis ergot 𝘂𝙬𝙎. If you look at all the options except the option A, all are arranged or all are belongs to one type, that is naxomica ergot cinchona datura. Most all are related to the alkaloid classification, whereas peppermint and clove, eucalyptus and garlic are nothing but they are having volatile oils in their proportion or the chemical contents or chemical classification, whereas digitalis and senna are cascara nothing but glycoside. I think this is a best example or best explanation. You can see right over here. Glycosides we had digitalis senna cascara, licorice, alkaloids are naxomica ergot cinchona and datura, and peppermint, low eucalyptus or volatile oils. That what we discussed. Clear? So in this type of questions, when we expect or when we see in that examination, 𝙂𝙎 examination, immediately look after that, which one is an odd one in the given examples or in the given options, so that you need to cross check all the options, then you can get the right answer. Yes, we'll go to the next question, that is the second question in this test series 2, that is the plants which are not differentiated into root, stem and leaves are. So, which plants are not differentiated into root, stem and leaves are? Option A plant Aegineans, Principes, Thallophyte and Bromeliads. The option 4 is Bromeliads. Tell me which one is the right answer. This question was given in the examination, 𝙂𝙎 examination 2020. Okay, which plants does not differentiate into root and stem and leaves? I think that is option C. Yes, that's the right answer. Thallophyte is the right answer. Where does not differentiate it into plants or root or stem and leaves. Okay, so these are the thallophytes. They doesn't differentiate it. They doesn't modify them into any of the root or stem or leaves. Okay, and Engler divides this thallophyte, especially this order into 13 phyla include they may include bacteria, algae, and fungi and lichens. Okay, all are thalafites. This thalaphite refers to that doesn't have or doesn't differentiate into root or stem or leaves. They exist as a specific form. Examples are 𝙂𝙂𝙎𝙬𝙬 and Cladophora. These are some examples for thalaphite. Clear? So that the thalapites are does not differentiate into the root or stem or leaves. Next, the third question in the CPAT test series 2 is a monoterpene or steroid compounds can be synthesized by which pathway? We know we have came across so many pathways. That's stigmic acid pathway option A, ornithine, mavolic acid pathway both A and C. Please tell me your answers. Yes. We'll see the right one so that we can go for the brief explanation of this option or this question. Option C is mavonic acid pathway, and this was the explanation. This was the explanation. Monoterpene steroidal compounds. Monoterpene and steroidal compounds can be synthesized by this mavonic acid pathway. Mavonic acid pathway. Steroid. You can look here itself in the steroids and okay, steroids, terpenoids are derived from this malonyl seaway is nothing but mavolic acid pathway. Clear? This is the briefing about the explanation of the third question. And fourth one, the cannabis and poppy latex belongs to which class? We know that cannabis and poppy latex are famous or belongs to which class? Option A is CNS stimulants, B analytics, C CNS depressants, D hallucinogenics. Option A CNS stimulant, B analytics, C CNS depressant, and D hallucinogens. Tell me the right answer. Yes, we'll see the right answer. Yes, hallucinogens because they create some hallucinations or they're directly acting on CNS. They produce some hallucinating effects. Okay. And so some of the examples for the drugs which are acting on CNS in different mechanisms of action. CNS analgesics are opium or morphine, stimulants are coffee, analeptics like naxomica and lobelia and camphor. CNS depressants are hyoscyamus and belladonna opium, where the hallucinogenics are cannabis and poppy latex. This is a poppy capsule where we made some incision with some needles so that it release some latex. That latex is collected and dried, and that is further processed into get cocaine, heroin, morphine, thebine, noscafen, and all these things. Clear? And this is cannabis. Okay. Yes, we'll move to the next question. That is the fifth question in the GPAT test series, which is given in the year of 2020-21 GPAT examination. Which one of the following is unorganized drug? You should answer this question very quickly because it is a very easy and fundamental in pharmacognosy crude drugs. A. Rauwolfia root, B. Acacia gum, C. senna leaves, and D. cinchona bark. We'll see the right answer. The right answer is acacia gum, which is not an organized one, it is unorganized. Why? Because organized one is having a cellular structure. So cellular structure containing plant parts are nothing but seeds, leaves, barks, woods, roots, rhizomes, flowers, fruits, and entire drugs which are having cellular structures are considered organized. We know that. Which doesn't have any cellular structures are considered as unorganized drugs. Like dried lattices, resins, dried juices, gums, and dried extracts. Okay, so that acacia is a gum. Okay, so comes under the gums classification in unorganized drug. That's it about the explanation. And now the next question in this GPAT test series, the sixth question is fruits which are derived from plants umbellifera all are the type of all of the type. In the gate 1991 this question is given. A cremocarp, B pericarp, C epicarp, and D mesocarp. Most of the umbelliferae family are having these types of carbs, which is the Which carpet does it have? We'll see the option or we'll see the answer so that you can understand. Yes, cremocarp. This is cremocarp. So all these structures, the two in indehiscent carpels, the two indehiscent carpels, that is one is dorsally and or laterally compressed. We know that dhania, right? That is coriander. The outermost ridges are considered to be the cremocarp. Clear? Yes. So this is cremocarp. All these are cremocarps. Okay. So the Cremoambellifery family are having cremocarp in their gross morphology of the fruits. Clear? Yes. Next, we'll move to that next question in this GPAT test series part 2. Who developed a homeopathic system of medicine? Nothing but who is father of homeopathic system of medicine? They ask. Option A, Hippocrates. Option B, Samuel Hahnemann. C, Galen, and D, Sedler. What's your answers? Yes. We'll check the right answer. Yes, we know that Samuel Hahnemann is known to be the father of uh, homeopathic system of medicine. We have so much of data regarding this, and he's known to be and he's the person Samuel Hahnemann, known to be the father of medicine, and he developed so many theories according to that. So he has some fundamental of principles, and he developed like principle based on his work. He developed this thing. Clear? Yes, we have came across of all these points in the previous class. Homeos, patho or pati, like and all this we have covered in the last session. Okay. Next question is meristematic tissues have following characteristics except we have permanent tissues and the meristematic tissues. But what are the characteristics except? So nothing but which one of the following is not a characteristic of meristematic cell? They do not have any capacity for multiplication. The cells are small and isodiametric in nature. Cells are thin walled with prominent nucleus, and the cells are compactly arranged without any intracellular space. Can anybody tell me the answer? Yes, good. But this question is appear in GPAT 2014. 2014. Okay, we'll see the option right over here. Yes, we know that they do not have any capacity for multiplication. They do not have any capacity for multiplication because this is a wrong option, which means meristematic cells have ability to replicate themselves or multiply, whereas permanent cell doesn't have the capacity for multiplication. So for the reason, it is a wrong option. Meristematic tissues have ability to replicate or to multiply. So for the reason, it is not a characteristic of meristematic cells. Yes. Yes, we can see the characteristics. These are isometric in nature. They consist of without any intracellular space. They are thin walled. They have vacuoles, and also they have a capacity to multiply. So that the first option said that it has no ability, but according to meristematic tissues, it is multiply. It has a capacity to multiply. So this is about the meristematic tissues explanation. Next question: Carbuncle is we have seen in the previous test series, that is different parts and then they function. Okay, but here they are asking about the arrangement or the nature of the part in the seeds. Caruncle, a watchy outgrowth from the microphyll, that is estro. Example is estrosexual growth from hilum covering the entire seed. Nutmeg. Outgrowth originating from micropyle and covering the seeds. Example cardamom. Enlarged funicle, that is colchicum seed. Colchicum seed. Tell me the right answer. Yes, the first option, that is Vati outgrowth from micropyle, that is castor seeds example. This slide we have covered already in the last session. Okay, Caruncle is nothing but outgrowth. This is the option. Okay, the Vati outgrowth of from micropyle as seen in castor or croton viola, that is Caruncle. Okay, next question in this GPAT test series. That's the two The unsaturated 2 carbon containing molecule which involves the argon absorption. Which of the following is having the 2 carbon containing molecule? Cytokinin or ethylene or gibberellic acid and oxygen. So if you have a basic fundamental knowledge about the chemistry itself, because our subject is pharmacognosy and phytochemistry, you can able to answer this question. Obviously, 2 carbons, ethylene, eth means 2, meth, eth, 2 carbons, ethylene. Okay, so ethylene has a property of or a function of argon absorption. So that's the reason ethylene, eth, meth, 2 carbon compounds. But apart from that, what the functions of ethylene we can check here: maturation and senescence. It promotes a flowering and fruit ripening, organ absorption, and response to biotic and abiotic stress and inhibitor of root elongation. Clear? Yes, most of the enzymes are by this ethylene. Okay. Next question in this GPAT test is: a compound now increasingly used as a standard practice for enhancing the flow of rubber latex by spraying onto the scraped bark of rubber tree, increasing the latex yield from 36% to 130%. This question will be, we have seen in 2018 GPAT examination. Brassinosteroids, acetic acid, ethopon, kinetin. We know that rubber latex is rubber trees. The from the latex, from the collection of latex only we are preparing, we are developing the rubber. But to increase the yield, which one of this thing is used? Let us see the option of this question. We know that ethopon. Yes, ethopon. Yes, these are the rubber trees. So the milky latex is collected in a bowl. So in order to increase this yield, we are spraying on the scrapped trunk part of the tree, that is ethopon, is sprayed on that so that it increases the yield up to 30 to 36 to 130% is ethopon. Clear? Yes. We'll go for the next question in the series. Quinol alkaloids are biosynthesized by which one of the following pathway? Quinol alkaloid, citric acid by tyrosine as a end product, citric acid tryptophan, citric acid phenylalanine, and citric acid cathinone. Which was the right option? We'll see. Yes, citric acid tryptophan, quinoline, alkaloids or quinine. Here itself, the precursor is tryptophan. If we ask or if we see that quinoline alkaloids are derived from tryptophan as a precursor, these are the examples for quinine, quinidine, cinchona, all the quinoline alkaloids. Clear? Yes. So this is a pathway where from tryptophan to other things. Clear? Yes. Next question in this GPAT test is polyploid is defined as polyploid. We know that the gate 4003 this question is asked. Okay, polyploidy means to addition of one chromosome or multiplication of entire chromosome set or submicroscopic change in DNA material or gross structural change. Which one is the right option? Yes, multiplication of entire chromosome set is a right option for this question, because a polyploid is nothing but multiplication, okay, of set of chromosomes. Okay, whereas if the particular thing is having a single set of chromosomes, that is monoploidy, more than one is diploidy, and more than two is polyploidy. Set of chromosomes is called as ploid, but if more than two are there, those are called as polyploidy. Okay, and yes, multiplication of entire chromosome set is referred as a polyploid. And for the polyploidy, there are some reasons or agents are there: X-ray, centrifugation, temperature, shocks, and other chemicals like colchicine, veterin, sulfonamide. All these causes the polyploid. Okay, but the colchicine alkaloid is the best known for its cause to cause the polyploid. You should remember, colchicine are the best example or best chemical to cause polyploidy. Clear? Yes. The next question in this GPAT test series 2 is biosynthesis of alizarin follows the which pathway? Whether citric acid, malic acid, both A and B or acid pathway. This question is given in 2015 𝙎𝑃AT. We'll check the right answer. Yes, both A and B. From the both A and B, 𝙂𝙀𝘋𝐼𝐴 acid and mavolic acid, it follows. It produces 𝘿𝙄𝙎𝑎𝑟𝑖𝑛. Okay. So from 𝙂𝙀𝑃 acid and mavolic acid, it can be produced. Yes. Sorry. By means of 𝘿𝙄𝙎𝑎𝑟𝑖𝑛. And we can see that A is derived from A ring is derived from 𝙂𝙀𝑃 acid and C ring is derived from the mavolic acid. Next, the most effective method for producing virus-free plants is root culture, meristem culture, floriculture, and somatic embryogenesis. In the plant tissue culture technique, it is an important thing. Tell me which one is the right option, my dear students. Obviously, the meristem tip culture can able to produce virus-free plants in the cultivation or in the plants. Okay. Yes. So meristem culture technique. Meristem, which means the shoot tip is collected, apical shoot tip is collected, and that is sterilized, and further it is regenerated to a virus-free plant as given in this procedure. Clear? We know this procedure and all these things. Okay. Yes. Next, the next question in this 𝙎𝙎𝑃AT test series is which plant hormone helps in breaking the dormancy of a plant, whether auxin, gibberellin, or cytokine or ethylene. We have this same discussion in the previous session also. Okay, which one is the right option? Option B is the right option. Gibberellin can able to uh, break the dormancy, and apart from other actions are rapid expansion of plant cells, stimulation of seed germination, breaking dormancy, induction of flowers, marked increase in stem elongation, increase in the size of leaves, increase of parthenocarpy leads to seedless fruits and seeds. So these are the functions of gibberellin. Next question in this 𝙎𝙎𝑃AT test series is in 𝙂𝙀 acid pathway, 𝘾𝙀𝑚ate mutase converts the 𝘾𝙀mic acid into option A, carotenoids, B, phytols, C, priphenate or prifenic acid, or D, gutta. We'll see the right option. Yes, prifenate. Prifenic acid is synthesized or converted from 𝘾𝙀𝑚ic acid with the help of 𝘾𝙀mate mutase enzyme. Okay, so this is an explanation or proof for the 𝘾𝙀𝑚ic acid is produced from or converted in the 𝙂𝙀mic acid pathway to from 𝘾𝙀𝑚ic acid to prifenate. The 𝘾𝙀𝑚ic mutase will convert the 𝘾𝙀𝑚ic acid to prifenate in the 𝙂𝙀mic acid pathway. Okay, so prifenic acid here, right over here in the box itself, whereas from 𝙂𝙀mic acid it gets formed. Next, in that question, 18th question of this 𝙎𝙎𝑃AT test series 2 is vitamin essential in tissue culture medium is option A, pyridoxine, B, thiamine, C, nicotinic acid, and D, inositol. Which was the right answer? Essential vitamin in tissue culture. We'll check the right option. Yes, B1, thiamine is the right option for the tissue culture medium. Yes, in the previous slide we have checked that pH of the thing. Now we are checking the vitamin which is required for the growth. Okay, we'll see the next question in this 𝙎𝙎𝑃AT test series 2. Which of the following is a biological method to control the pest? Option A, crop improvement, B, pathogen destroyed with another living organism, C, crop rotation, and D, plugging the field deeply. So to control pest, which one is the best method? Yes, pathogen destroyed by another living organism. In order to kill rats, we uh, use cats, dogs, we domesticate them. Okay, as like that. To which one is the biological method to control pest? Okay, nothing but living organisms that we are using. And yes, we have different techniques or methods of control, pest control. In that, agriculture method, biological method, chemical method, mechanical control is there. Yes, that's about the explanation of that thing. And now moving to that, Who is known as father of medicine? Can anyone tell me the option for this question? A. Galen, C. Sidler, and C. Hippocrates, and none of the above. Can anybody post the answer? Yes, my dear students. Let us see the right option so that we can go forward without wasting time. Hippocrates is known to be the father of medicine. We know that he is an ancient scientist and is rooted to be the father of medicine. Okay, Hippocrates. And there are some of the scientists and their, you know, the fathers of various fields. Hippocrates is known to be the father of medicine. Aristotle is known to the natural science. Galen, first pharmacist, and Shushruta is known to the father of Indian surgery. Theophilus is father of botany. Okay. Yes. And the next question, 21st question in the GPAT series 2 is pharmacognosy is an outgrowth of where it is originated, whether from Atharvaveda or Maitreya Medica or Pharmacopoeia or Charaka Samhita. Every part or every option is different. Option A Atharvaveda, B Maitreya Medica, C Pharmacopoeia, and D Charaka Samhita. What is the right option? Maitreya Medica is the right option because from the Maitreya Medica only it is originated. Okay, so that's an explanation. From this Maitreya Medica, pharmacognosy word is explained or is developed. Yes. And the next question in this GPAT test 2 is callus is an organized mass of cells, an unorganized mass of cells, an organized mass of nucleus, and unorganized mass of nucleus. Which one is the right option? Don't worry, we'll take the right option and we'll have a clarification and explanation too. Yes, option B is right, an unorganized mass of cells. According to the statement or definition, callus, the unorganized, undifferentiated mass of plant cells referred as a callus. Unorganized, undifferentiated mass of plants are referred as callus. Clear? Yes, these are callus. And 23rd question in this test series is atropine biosynthesis involves which of the following amino acid? Option A ornithine, tyrosine, dopamine, and tryptophan. We'll check the right answers. Don't worry, my dear student. Meanwhile, what's the answer? Ornithine is the amino acid which involves in the atropine biosynthesis. We'll see this explanation. Okay, in that we have seen tryptophan, we have seen, and now we have seen that ornithine, the first one, which includes in the development of tropane alkaloids. Okay. Yes. Let's see the next question. Cannabis poppy latex and belongs to which class? Already we have discussed the first slide. That is, they are belong to hallucinogens. Okay. So these are the some of the examples with respect to the action on the CNS. Okay. And last but not least, which of the following growth regulators induce embryogenesis? Embryogenesis. Option E auxins, option B cytokinins, C gibberellins and abscisic acid. Can everybody tell me? Option D is right. It is an abscisic acid which induces the embryogenesis. Clear? Yes. So these are some examples of plants and the requirements in the culture techniques. Clear? Yes. Yes, my dear students, this is for today's session and this is for this GPAT test series 2. As you all know that in order to get such type of content and test series and mock tests and other live classes, you need to have or you need to download our app called GDC Study Center or GDC app. You can directly go to the Play Store and you can search it GDC Classes app and you can download from it. Clear, my dear students? I think it is interesting and a useful session for you all. Yes, I hope you enjoyed the class and have a great day. We'll see in the upcoming test series classes until you reach your goal, until our examination comes here. Clear? Yes, my dear students, have a great day. Thank you.